Hello everyone and welcome to this audio podcast today. This audio podcast is part of the Serology and Squil. This audio podcast is part of a Hustle and Stereotype. This audio podcast as part of a Defcon premiere, and this audio podcast as part of a Storm Robotics production. So continuing on and looking at transplant immunobiology and idiosyncrasy. So conversely, CX3 CR1 knockout laboratory mice or murine knockout laboratory investigations wherein recipients were treated with a subpharmacotherapeutic dose of early generation IST pharmacotherapy, and wherein so for example CSA or cyclosporin A, and where CSA accepted cardiac allografts CSA accepted cardiac allografts accepted allografts additionally were evidenced as having longer than CX3 CR1 laboratory mice or laboratory wild type mice allografts and allograft prognoses, and the process of leukocyte trafficking from peripheral circulation into tissue spaces involves a series of interactions between soluble mediators and surface molecules expressed by the endothelium and leukocyte, as well as subsequent interactions with extracellular matrix. Chemokines mediate events at multiple stages in this process. Investigations have also looked at urinary levels of CXCL10, looking to understand how such chemokines elicit or mediate acute allograft rejection, or AAR. CXCR4 present on T cells and B cells, monocytes, neutrophils, and blood-derived dendritic cells and others is the most broadly expressed chemokine receptor. CXCR1 and CXCR2 are expressed on most leukocytes, but also appear to be functionally significant only on neutrophils, monocytes, and macrophages and mast cells. CXCR3, CXCR5, and CXCR6 are expressed or have been evidenced as being expressed on cells of lymphoid lineage. CCR1, CCR2, CCR4, CCR4 and CCR10 are expressed mainly on lymphocytes, monocytes, and monocyte derived dendritic cells. CCR3 has or have a unique expression and is identified or is found on eosinophils, mast cells, and basophils. T helper cell type 2 lymphocytes and certain dendritic certain dendritic cell populations are also or have also been identified or established in as part of the unique expression aligned to CCR3. That draws this audio podcast to a close. This audio podcast as part of a Serology and Squil. This audio podcast as part of a Hustle and Stereotype. This audio podcast as part of a Defcon premiere. And this audio podcast as part of a Storm Robotics production.